Dear students and participants, so in this lecture we'll discuss about bioprinting of lung tissue. Before that, we try to understand what is the need of bioprinting a lung tissue. So, as you know, there are many patients who suffers from end-stage lung diseases, and many of them actually require transplantation, like where that lung tissue collected from donor can be transplanted back to the patient, so to save this kind of end stage lung disease patients where they are actually where their actual lungs or their own lungs does not function. So in that case the transplantation is very much required, but for doing that kind of transplantation, highly specialized expert surgeons are required. So that is one challenging thing, because the number of highly specialized expert surgeons are limited, and but for doing this kind of lung tissue surgery or lung transplantation, that kind of expertise is required. And we'll in the later part we'll discuss why this kind of surgery can be very much complex. The other problem associated with lung tissue transplant is the, like with other tissue, other, as we have discussed for other organ or tissues, like the risk of rejection and also the limited supply of liver transplant, because there are limited number of donors, but the need for transplant is way higher than the actual supply. So that's why these are the major challenges, like the requirement of expert surgeons, risk of rejection, and donor scarcity. There are various attempts by which there are various attempts to fabricate or to regenerate in vitro lung tissue model. We'll discuss some of these things in this lecture, but still now a 3D alveolar model, like that alveolar is the highly complex microvascular network structure that is present in the lung for effective gaseous exchange. That kind of structure has not been successfully fabricated because of the complexity and the dimensions of the alveolar structure. If you see, the native lung tissue has an intricate diverging network of airway with alveolar sacs that are up to, yeah. So native liver tissue, they has a intricate diverging network of airway with alveolar sacs. There are multiple, or there's also multiple alveolar sacs present. So those sacs are actually 200 micrometer in diameter. So this 200 micrometer diameter alveolar sac that is present in number of these sacs are present in the lung tissue. So as I was mentioning, that native lung has an intricate diverging network of airway, and each alveolar sacs, they are almost like 200 micrometers in size, and because the multiple presence of multiple alveolar sacs, the total surface area to volume ratio is very high for this intricate structures. It's almost up to 100 meter square, and because of this, there is a efficient gaseous exchange. So because the surface area is very high, so the gaseous exchange is also very efficient in case of lung because of this alveolar structure. And in this alveoli, when we see further the microstructure of alveoli, there is a delicate blood air interface present, where the branching vascular networks that divide into capillaries and adjacent to the thin respiratory membrane outlining the alveolar sacs. So what happens here? There is a membrane, respiratory membrane, where this the alveolar sacs are placed. Alveolar sacs is present where there is a presence of this thin respiratory membrane, and the other side of this thing, the vascular networks, a capillary network is present within this thing. So that's why the gaseous exchange can happen from the air, from that air within the alveoli, the air presence of air, and the other side the blood. So that air and blood, that interface, because of that the gaseous exchange happens. So this average thickness of this respiratory membrane, that is very fine, like zero point 0.5 micron. So that facilitates the oxygen and carbon exchange at 5 liter per minute of blood in the pulmonary capillaries. So at that high speed, when the blood flows in, so in a minute, 5 liter of blood can be passed through this network, and can get as gaseous exchange can happen, where the oxygen flow will diffuse from the air to the blood capillary area, and carbon dioxide can again reverse. Carbon dioxide can diffuse from the blood to the air space in the alveoli. Now, because of this intricate, complex structure, reproduction of this tissue is highly challenging, and till now, there has been several attempts to regenerate that whole in vitro lung tissue, but functional in vitro 3D alveolar model has not been successfully fabricated. There are few instances or few reports where uh, researchers have tried to fabricate a part of that alveoli respiratory structure, but the whole thing is still not been done. Now we'll discuss what are the different strategies for liver fabrication. If you see the liver, as I mentioned, that the liver has a very complex microanatomy, so that's why people started reproducing or replicating that and microanatomy of the structure using micro fabrication techniques like lab on chip or microfluidic device. So those things are mostly used for fabricating that microvascular network and air, and that respiratory membrane like structure. So and other side air is present, so that the gaseous exchange can be studied, and all these things can happen. So these kind of studies are present, but it is very difficult to fabricate a device for where uniform microchannel formulations and seamless connections are there, because for requiring a device where if we have to create a whole this micro anatomical this structure, this liver alveolar structure, so it is the requirement is like uniform microchannel formulation and seamless connection. So that is very challenging, but several groups have also been explored the whole organ dissolution approach. So what happens here, because there are there are some reports where people have tried to dissolution the whole lung tissue using detergents and others, there are several other chemicals. So in that process, the main thing is the liver, the whole organ is first miss the sorry the liver with that the vascular network. Suppose the artery and the capillary artery that is supplying the blood to the thing. So that is connected to the perfusion pump, and through that thing the detergent solution is being pumped through that one, and because of that what happens, the that detergent can remove the cells. So that process is called dissolution. By this way, the cells can be removed from the whole lung tissue. The remaining thing is the extracellular matrix component within that branching within the typical microarchitecture that is still there. That will be still there present there, but the cells will not be there. So that is the dissolution lung tissue. Followed by that, the cells. The cells typically the cell tiles that is present within that particular tissue that has been seeded back to that dissoluted tissue. so that if we can get optimum cell seeding within that structure, so where, like suppose in the lung parenchyma, the lung fibroblast, and also in the smooth muscles, other things, those cells are, they seeded, and in the that blood vessel channels, so there are vascular network there, the endothelial cells are seeded there. So by this way, the typically whatever the cells are present in that lung tissue, those cells are seeded back, and then you can again, that tissue, it has been shown that this kind of digitalized liver, whole liver tissue, when it is the cells are seeded back, they can function like the native tissue, where because the main complex microstructure network is still present there, and the microvascular network is also still present there. Because of that thing, the structure can again start again. If we can successfully seed the cells, or able to seed the cells, then that can function better. But here the major challenge is the cell seeding, because when we destroy the tissue, the extracellular matrix is actually, there is a little contraction of the extracellular matrix, that the whole structure, where because there is another thing, because there is no porous open porous structure present in that the tissue, because of that thing, because of that, the cell seeding is very challenging. But if the cell seeding can be very well done, then this technique is very powerful. But the major bottleneck is the cell seeding in that case, I mean decentralized tissue approach, decentralized whole tissue approach. But so that's why this process has not been very successful, but people have been trying to use that for various applications. The other thing also, the few groups, they have tried to devise a lung assist device, means that they tried to design a lung assist device where the that gaseous exchange, suppose the gaseous exchange can happen with the blood flow typically, uh, and so that the gaseous exchange can happen, and the oxygenation and deoxygenation, carbon dioxide, those things, those process can be done. So that kind of implantable lung assisted device also have been fabricated, so that can augment the lung function, and that can also act as a bridge to transplant or possible destination therapy. Like many people, as we have discussed earlier, like there are many patients who are awaiting for lung transplantation. So they can be, for them, this kind of lung assist device can be very helpful, where this kind of things are there. So we'll discuss some of this in the next slide. So the first one is the lung assist device technology with physiological blood flow. So this was done by Hoganson et al. They have fabricated, using a biomimetic design principle, they have fabricated a microfluidic vascular network for blood inflow from the pulmonary artery and blood return to the left atrium. So because in our body, the blood, typically the deoxygenated blood that comes through the blood comes through the uh, pulmonary vein, or pulmonary artery, and that artery takes the blood to the lung, and then after that lung, that blood goes to the left atrium. From there it goes to the left ventricle, and from there it goes to it goes to the whole body. So the for because of this oxygenation process, so that's why. So now what they have done, they have created a microfluidic vascular network, where that can be connected to the pulmonary artery, so that the blood from the pulmonary artery that goes to the vascular network, and then the gases, they also used a respiratory membrane, using a silicon membrane was used as for this, to act as a respiratory membrane, so that can be present in between this vascular network, and the other side there is a channel for the air passage, so that the gases exchange can happen through the silicon membrane to this blood, and then the blood, after this gas exchange, the blood can return to the left atrium. They have also done a computational fluid dynamics analysis, they have used to optimize the blood flow within this vascular network. So by using this computational fluid dynamics, they studied the what is the flow rate, is it how much flow, what is the optimized flow rate, optimum flow rate, so that the gaseous exchange can be fine. Then also they have used a micromill variable depth mold because the mold they have micro milled, and with 3D features was created then to achieve the both physical blood flow and shear stress. So that also they have created a mold for 3D features to again to devise this kind of structure. So the gaseous exchange can occur across the thin silicon membrane that is present between the vascular network and the adjacent alveolar chamber. So in their devices, there is a on the top there is a alveolar chamber, then the silicon membrane, and the below there is this vascular network. So the silicon membrane can be used can represent the respiratory membrane, and the vascular channel and the air channel both are present, and so that the gaseous exchange can happen. So now this kind of device, and then they found that a physiological gaseous exchange can happen, right? So whatever this, like the general typically hollow fiber oxygenators are being used to oxygenate the blood in case of means the people who are almost like end stage lung diseases. So where lung assist devices are basically typically used, where this lung hollow fiber oxygenators are used to oxygenate the blood, right? So they have found that their device can function similar to the this hollow fiber oxygenator, but they can maintain a physiological blood flow. That is very important. By maintaining physiological blood flow, they can the gaseous exchange what they have achieved, that is almost typically similar to the hollow fiber oxygenators. So this device can be scaled up further to create an implantable ambulatory lung assist device, so that can save many patients. And if some patients when they need this kind of lung assist device, they can use that device. So this is this. Please go through this literature. Due to copyright issue, I have not reproduced or used any images from this particular paper, but please go through this literature. You will for bit further reading and further understanding. The next study that I'm going to discuss is where a organ level lung function has been reconstituted on a chip device. So this is also another a, micro, a in a chip scale a device is created where it's a uh, that can represent that they can reconstruct the organ level function. So here a biomimetic microsystem was created, so that can do the critical alveolar capillary interface of the human lung to reconstitute that critical functional alveolar capillary interface structures that they have done in this study. And this study was published by Hu et al. In Science Journal. And this bio-inspired micro device that reproduces the complex integrated organ level responses to bacteria and inflammatory cytokines introduced into the alveolar space. So what they have done here in this work, they have created a bio-inspired micro device, and where they could see that how suppose the bacteria when the bacteria goes inside the airway tract, then what it does, how it will respond, how it will interact with the lung tissue, suppose the airway tract. So that kind of, because in this case they have developed a airway tract, so how this bacteria can interact with the airway tract, that kind of response also they could achieve within that micro devices, right? And this device also mimic uh, the inflammatory cytokine responses. Suppose when in the alveolar space, this device also has a alveolar space and also has a or one the blood one the another vascular space, right? Both these things are present, and in between there is a fine membrane like structure. Please go through this literature for the images and other things. I have not reproduced or used the images because of the copyright issue, but please go through this literature for further understanding. You will see that they have created it for a two chamber device where one chamber is used for the airway passage and another chamber is used for the blood vessel supply. Suppose that for the blood vessel supplies to mimic the blood vessel, suppose for the liquid for the fluid supply, and the other there are two side channels also by which if we, they, when they use that they use those two side channels to apply vacuum within this thing, so that the membrane can be stretched and strained, so the strain can be created on the membrane. So because they want to achieve that, they want to create the physiological response in that device. So that's what. But when they found that when they introduced inflammatory cytokines within that can also, the response is also very similar to the organ, native organ level responses. Other thing also they have, as I mentioned, that because through the side channels, they applied vacuum so that that membrane can be strained. So this lung can mimic the cyclic mechanical strain. Because of this, that can accentuate the toxic and inflammatory response of the lung to silica nanoparticles. Now when they also introduce silica nanoparticles within the alveolar space, they also find that uh, that kind of toxic response is kind of generated because of this, the presence of this thing. Other thing is the mechanical strain also they have used to enhance the epithelial and endothelial uptake of nanoparticles and stimulates their transport to the underlying microvascular channel. So these kind of things also they have done, and they found the similar effect to the physiological breathing on nanoparticle absorption are observed in whole mouse lung. Suppose if a silica nanoparticle is being administered to the mouse lung, the response, whatever the response we can observe, the similar response they could see in their devices. So this kind of, so this study, in this study, the importance of this study, they have reconstituted a alveolar capillary interface structures, and then they studied the different physiological functions. Suppose the responses, interaction with the bacteria, inflammatory cytokines, in interaction, if they introduce some inflammatory cytokines, then what is the response of this thing in that alveolar shape. Also this kind of lung can mimic the cyclic mechanical strain. If the cyclic mechanical strain is applied to this device, then that how it will respond to the, it will respond to the structure. Those kind of things they have been studied within this within, they have studied those things within the device, within this kind of device. Other than this also, when they apply stretch and strain, then that epithelial and endothelial cells, they can, their uptake of nanoparticles is also get stimulated. So that is typically that kind of response in our, in the not native lung physiology, native lung tissue or the native airway tract. Also we can see similar uh, similar similar observation have been made before. So that is the importance of this study. So please go through this literature for the further understanding. Let us now discuss about another very important study where an in vitro air blood barrier was recreated using inkjet bioprinting technique. So here an in vitro, as I said, an in vitro human lung basement membrane model was fabricated using inkjet bioprinting technique. Inkjet bioprinting, if you recall, then we have discussed this before, that this technology works on the principle of generating droplets. So these droplets can be patterned on a substrate or a supporting structure, and then this the also cells or any other bioactive molecules can be incorporated within the inkjet bioprinter. No, sorry, within the biowink, where a water-like viscous material like either media or cell culture media or buffer can be used. And in the printhead there are hundreds of nozzles lined up, so at a time we can generate hundreds of droplets. So that's why this technology is high throughput method for fabricating of any particular structure. But a support structure or substrate is required to generate 3D model. So in this case, what they have used an inkjet bioprinting technique, and also they have used two different types of cells like endothelial cells and the alveolar epithelial cells. So these two cells represent the alveolar and epithelial structures. Sorry, alveolar and the blood vessel structure, vascular structure. So what they have, and they have also used a material that act as a basement membrane, like the matrigel they have used. Matrigel is a material that's also a extracellular matrix component that has all the extracellular matrix component present in a particular tissue. So that's why they have used this matrigel for to generate the basement membrane that is present in between the alveolar and the blood, that barrier, or in this the membrane, uh, the lung, in the lung tissue there is a membrane, always there is a membrane present in between these two tissues. So they found that the manually seeded cells, also they have another thing they have done is they have compared both bioprinted and manually seeded cells for fabricating this kind of structure. So we'll discuss this, how the about the fabrication, how they have used the inkjet printing for fabricating the structure. But they have compared also, they have manually seeded both these things. But what they found, when they manually seeded cells, they found a patchy layered clusters with thick ECM between the cells that hindered communication between the cells. When the manually cells are seeded, then what happened? Because in case of manual seeding, there is no control over positioning of the structure of the cells into a particular layer, into the particular position of the substrate. That's why the cells can form a patchy layered cluster, and a thick basement membrane can be present between the endothelial cells and the epithelial cells. So there is no communication between these two cells if the because of the gap is larger than the critical size. Alternatively, they also found that when the cells were printed, they formed a uniform surface of they form a uniform layer on the surface of the membrane, and that so that a very small gap between the two cells where the basement membrane is present in between them, that is that form they can allowing the communication between those cells. So thus bioprinted monolayer culture demonstrated enhanced barrier quality than the manually formed samples. That is the uh, conclusion from this study. So let us now discuss, let us understand how they have fabricated this device. So what they have done here, at the first day, a matrigel, one layer of matrigel is printed with the inkjet bioprinting technique, like here. So a very fine layer of matrigel is printed day one, and then subsequently the matrix, they have allowed the material to get solidified or cross-linked, and then they have again bioprinted the endothelial cells suspended in the cell culture medium, like here. Bioprinted cells are bioprinted on top of that matrigel. So here endothelial cells, they have used EA.HY926, a endothelial cell line they have used. That is the second layer printed, and then they have given a gap of 2 days for these endothelial cells to get settled down on the matrix, so that they form a nice monolayer structure. And then they have, in the day 2, they have printed another layer of matrigel basement membrane on top of this endothelial cells, this. And then subsequently the epithelial layer cells, those are also suspended in cell culture medium. They were printed on top of the second matrigel layer. So here A549 cell lines they have used. These are alveolar epithelial cell lines that they have printed. Now, so here what do you see? We get a epithelial layer and endothelial layer, two different layers, and in between the a layer of matrigel is present. So matrigel is acting, so here that is acting as a basement membrane and separating these two cell lines, cell monolayers. So this is the engineered two-layer culture system. So then they have cultured the whole thing inside inside the media, where all they have provided all the necessary conditions for the maturation of this thing. Similarly, they have also the same process they have followed, but but they have not bioprinted but manually seeded the cells on top of the cell. So suppose at the first the material was printed, then on top of that the endothelial cells were seeded at day 2, and then again at no sorry material layer was first seeded, and on top of that endothelial layers were seeded, and then they have waited 2 days for this thing for endothelium to grow and form a layer for monolayers, and then they have printed the another layer of material, and then on top of that the epithelial cells were seeded, and then they have cultured that something. So two different strategies, one manual and one bioprinted, they have used here. Now let us see the difference between these two techniques, whether is there any difference in outcome. So here they have what they have found. If you can say this column is for manually and this column is for printed. So here you can clearly see that when the cells are manually seeded, they do not form a continuous monolayer structure. But in case of printed, the cells, endothelial cells, they form a continuous monolayer structure, where the green color is the F actin cytoskeleton and the white thing is the red nucleus, cell nucleus. Both endothelial cells and epithelial cells, both cases you can see that when the cells are printed, that time they form a continuous monolayer, but in this case they not. Also here they have shown the thickness of the construct, and in this case if you see the thickness is more than one monolayer, but in this case a perfect monolayer is formed when the cells were printed. Again they have characterized this by, that means where they see the manually and the printed. If you see the printed cells, they formed a nice model continuous structures, but manually that is not even can. And the cross section will be also showing the same structure. In this case, a they have a further they have shown the magnified version of this thing, and here it can be clearly seen that the cells here, the cells are labeled cadherin B cadherin, the pink, that is typically present on the cell surface, but because that is the cell surface marker. When the cell to cell communication tight junction is formed, then the V-cadherin cells express this V-cadherin. So the V-cadherin we can clearly see this. In this case, the V-cadherin expression is higher than the other one, and the cell form a typical monolayer thing, and the endothelial cells are labeled with green color. So this is the thing, and then again also they have characterized this by histological staining, and in the histological staining it is clearly shown that the collagen here, the collagen fibers are marked as blue color, the cytoplasm is stained red, and the cell nucleus is dark brown. So here you can clearly see in the case of manually a typically patchy, thicker matricial structure with the cells. The cells also they are distributed randomly, and there is a multi-layer structure is formed. But when it is printed, a monolayer culture of the is typically formed, and so in this case a particular order is maintained. So typically definitely, so in this study, from this study we can understand that bioprinting technique can be very well used for creating this kind of barrier structure where the cells are present across the basement membrane. Now let us understand what are the future scopes of all this work. If you see all these different works, whatever we have discussed, they all created some part of the lung structure, like either the alveolar and vascular barrier structure, or the alveolar blood air barrier structure or membrane structure, or even the lung lung tissue lung tissue structure, lungs, a part of the alveolar structure. So these structures are they are very small. So further advancement of these technologies are is required for so to develop a fully functional lung alveolar structures, or even the airway structures. Other thing is some of the techniques they consider the vascularization, but a detailed, intricate, complex vascular network is very important to establish that the blood air barrier structure, blood air structures, various structures where the gaseous exchange can happen, and we can study those things. And even to generate a fully functional lung tissue, the intricate vascularization is very much required, as we have seen before, that in case of lung, the vascular structure, that they form capillaries, intricate capillary structures, and that capillary actually is is and is creating adjacent to the that adjacent to the basement membrane, and to the other side there is this epithelial cells, or the air that structure is present where the air is getting circulated. So that because of that, there is a gaseous exchange can happen. So vascularization is very important for this thing. So as we have discussed various vascularization strategies, probably some of these strategies can be implemented to generate this vascularized tissue structure. Other thing is the cell culture has to be optimized. We can, as we have seen, that when the cells are basically plated or seeded, they cannot form a ordered structure. But if the cells are when the cells are bioprinted, they can form a ordered structures. So, and we need to use certain cell culture techniques. Probably we need to come up with better cell culture techniques to optimize this thing. Other thing is the scaling up of all these technologies, because this most of the technologies, they can create a small structures or building blocks mostly. But if we can integrate all of these, and then we can scale them up, then probably we can print a whole tissue structures. So the finally ultimate goal of this all these things is to develop a bioprinted lung tissue for transplantation. And definitely if that is possible, then it can save many patients who are actually awaiting for transplants, and who are also who are mostly, and they have this end stage diseases. And and then we can save many more patients if we can develop bioprinted lung tissue for transplantation. So with this, we come to end of this uh, bioprinting of different tissue, or bioprinted of different organ structures. So we'll start with a new topic in the next lecture. Thank you very much for attending this lecture. We'll see you again.